back to Miss B Biology. This is a revision lesson on the required practical for photosynthesis. So grab your book and grab your pen and let us begin. The aim of the required practical is to investigate the effect of light intensity on the rate of photosynthesis. Step 1. Cut a piece of pondweed and place it in a boiling tube that contains sodium hydrogen carbonate solution. Students ensure that the cut end faces upward. The sodium hydrogen carbonate solution releases the carbon dioxide that is required for photosynthesis. Let us look at the equation for photosynthesis. It is carbon dioxide plus water in the presence of light energy to produce glucose and oxygen. So the sodium hydrogen carbonate solution will release the carbon dioxide that is required for photosynthesis. Step 2. Place a light source 10 centimeters from the plant. Students, if you're using a regular light bulb, the ones that give off quite a bit of heat energy, you need to place a beaker filled with water in between the plant and the light source. The water will absorb the heat energy that is coming off so that it does not affect the temperature of the experiment. We are using an LED bulb for this required practical though, so we do not need that beaker. Next thing that I want you to do, students, is to leave the experiment there to acclimatize to the light intensity for about 5 minutes. Step 3. Set a timer for 1 minute and count and record the number of bubbles observed. Students, remember now, the bubbles coming off is as a result of the oxygen that is produced in photosynthesis. This is what we're using to measure the rate of photosynthesis. At the end, students, repeat 2 more times and calculate a mean. What do I mean by that? Set the timer again and count the number of bubbles coming off for 1 minute. You do that 2 more times, as I said before, and calculate your mean. Next thing that you'll do, students, is to move the pond wheel 20 centimeters away from the light source. Leave it there to acclimatize to the new light intensity. Then you set your timer for 1 minute and count and record the number of bubbles observed. And again, you'll repeat 2 more times at this distance and calculate your mean. Next step, move the pondweed 30 centimeters away and do the same thing. Do it for 40 also and 50 centimeters. With the data you have acquired, you should be able to draw a graph, mean number of bubbles against distance, and the graph should look similar to this one. So let us now analyze this graph. First point, the further the pondweed from the light source, the lower the light intensity. That makes sense. The further the pondweed from the light source, the lower the production of oxygen. Students, remember now, the bubbles formation is as a result of the oxygen that is being produced or the oxygen that is given off. So let us analyze point number 2. It says the further the pondweed from the light source, the lower the oxygen production, right? So if we're looking at, say, this distance right here, this is the amount of bubbles that would be produced. And if we go further away from the light source, right, we notice that number of bubbles decreased right there and decreased even more right there. So point number 2 is correct. The further the pondweed from the light source, the lower the production of oxygen. Last point. This means that the further the pondweed from the light source, the lower the rate of photosynthesis. Let us now look at the variables of this practical. So you have the independent variable, this is the variable that is changed, the dependent variable, the one that is measured, and the control variables, the variables that must remain the same throughout the practical. So what was changed in this practical? What is the independent variable? The light intensity. Remember now, students, we moved the lamp from 10 centimeter to 20, right out to 50 centimeters. And the further away we move the lamp from the plant, the lower the light intensity, right? So light intensity is the independent variable. The dependent variable is the number of bubbles that we counted per minute. And later on, I'll show you how you could measure the volume of oxygen produced. The control variables now, these are the ones that must remain the same, and we have quite a few. The first one is temperature. We control the temperature. That is why I said to you, if you're using a regular bulb, you must place a beaker with water in front of the plant so that the temperature of the experiment is not affected. Next one is time. Remember all of the repeats that we did, we kept it at 1 minute. We counted the number of bubbles for 1 minute. Next one, the volume and the concentration of sodium hydrogen carbonate must be the same. And lastly, size and species of the pondweed must be the same for all your trials. Let us now look at the improvements for this required practical. Instead of counting the number of bubbles, students, you could use a gas syringe to collect the volume of oxygen produced. Because when you're counting the bubbles, students, if the bubbles are moving very fast, you could miscount. And also the sizes of the bubbles, they're different, and so it might not be accurate. So this is how you would set up the experiment with the gas syringe. Very similar to how you set it up first, but instead of counting bubbles, you would attach a gas syringe and the top bit must be sealed very tightly, students, airtight. And then you would read the volume of gas from the gas syringe. Another way we could set up this experiment is to place the pondweed inside an inverted funnel. Place the funnel in a beaker filled with water and then place a measuring cylinder filled with water over the funnel to catch the bubbles. As the bubbles rise, what is going to happen? The water will be displaced by the gas, and then you can go ahead and measure the volume of gas collected. I hope that this was clear to you, students. Thank you so much for watching and all the best in your exams. Bye bye.